Hi guys, I hope your week has been well. And I am Quinlene, your biology tutor at the San Antonio Math Include. And I had taught a lesson last week, but if you couldn't make it, that's okay. There is the slideshow material that will be able to be shared with you guys if you guys would like. And just please email me. It's kunling.wu and then at outlook.com. So unfortunately, I won't be able to make it to the Saturday's meeting. I'm very sorry because I'm on my school's varsity volleyball team, and we're approaching the end of the season, which is why all the schedules and stuff get kind of messed up. Because there's unannounced competitions on weekends. I will be gone for the entire day on Saturday, practically. However, I'll be covering all the material that will be needed to cover this Saturday. We will be learning Chapter 4, Cell Structure in Biology, and this is a very important topic to know because cells make up life, basically. So, cells are the fundamental units of life, in a way. The cells are the building blocks of all organisms. So, whether it's like a small bacteria or like a bird or even like yourself included. So, the cells in single cell organism, the cell is basically everything that they have. A single cell organism is simply a cell that can survive on its own. However, in multicellular organisms like human beings, the cells work together and there are also like a lot of different types of cells. So multicellular organisms are organized in a hierarchy. So the cells are the basic unit, and a bunch of cells, when put together, they form tissues. And the tissues, the reasons why the cells are put together in tissues because they perform a common function. And when you combine several tissues together, you form an organ, such as your stomach or like an intestine. So organs working together make up an organ system. For example, your digestive system, your neural system, your nervous system. So multiple systems that function together form the entire organism. So in your body, you have the neural network, the nervous system, digestive system, your cardiovascular system, circulatory system, etc. And you're only surviving because you're relying on all these different cells, which make up of all these different like higher hierarchies of parts. So the cell size varies by quite a lot, and most are actually too small to be seen by the naked eye, which is why we have to use microscopes to make the small cells easier to see for us. So there are two very important concepts to know regarding how to use a microscope. The first one is magnification, and then the second one is resolving power. So magnification is the process of enlarging an object in appearance. It's basically like holding a simple magnifying glass to a book and you see the letters in the bigger version, but the same with organisms such as this butterfly over here. You can zoom in on it until you see this little fur on the butterfly. However, there's also the parameter of resolution. So resolving power is basically the ability of a microscope to distinguish two adjacent structures as separate. So instead of seeing the structures as all melted together and jumbled up like the one on the left, if we have high resolving power, we'll be able to see that this is actually made up of different little hexagonal structures. So the higher the resolution, the better the clarity and detail of the image. And the resolution and the magnification are all adjustable in the microscope. So microscopes with different optical systems produce images for different studies. So the most common microscope that most schools use, my school included and probably your school as well, are the light microscopes, and they work relatively simply compared to other microscopes. They basically bend visible lights to provide magnification, and they send the light through special lenses to reflect it around and to magnify object. However, if we're using light microscopes, if we're looking at transparent objects like cells, which naturally do not have color, they must be treated with chemical stains, such as this green stain over here, this blue stain, sometimes like pink or purple stains, to distinguish the different parts. However, the electron microscopes also exist. There are a relatively new development, although we've been using them for the past several years. There are of course like even better microscopes, but they're super expensive and they require a lot of like I would say money resources put into them to be able to afford them. But electron microscopes achieve higher magnification and resolution using beams of electrons. They're very expensive, and how it works is that they basically send a very tiny needle, and it sends electrons across the surface of whatever thing we're checking out under the microscope. So for example, in this blood cell over here, by sending the needle and sensing all the electrons that got reflected off the surface of the cell, we actually are able to make a diagram of the cell. So transmission electron microscopes can show fine detail within cells, and scanning electron microscopes provide 3D exterior views such as over here. If you Google some scanning electron microscopes images, they are actually super cool, and I highly recommend doing that. And I believe here we're looking at some blood cells. So the cell theory is the underlying principle of biology. It is what biology is about. So there's 3 parts to the cell theory that you must know. First one is cells are the basic units of life, and then all organisms are made of cells. And third, all cells come from pre-existing cells, which means I can't just have nothing and then a cell just pops up. Like, this cell must come from something. And this is also why a lot of scientists even now are debating about where that very first cell comes from, because if all cells come from other cells, then, what happened? How was the first cell created? So all cells have 4 common components no matter what kind of cell they are. They all have an enclosing plasma membrane, which separates the cell's interior from the environment. It's basically this little wrap around the entire cell. The cytoplasm made of cytosol, which is an aqueous environment in which the other components of the cell are found. So as you can see, the dotted region here is the cytoplasm. It's kind of like a gooey gel kind of environment, and there are a lot of other components of the cell floating around the cytoplasm. There is the DNA, which is genetic material of the cell, and we also have ribosomes, which synthesize proteins. I will be able to cover all these in the rest of the slideshows. So first of all, let's take a look at prokaryotes. Prokaryotes are organisms in the domains archaea and bacteria. Archaebacteria, which is the domain archaea, they're quite different from bacteria. In fact, they're very tough organisms. They usually live in extreme environments. However, you might be more familiar with bacteria, which are usually seen as the causes of disease. They're very tiny usually, and we need microscopes to look at them. So they're all prokaryotes, which lack a membrane-enclosed internal compartment, such as the nucleus. In this case, their DNA, the prokaryotes' DNA, are just floating around in the cytoplasm. And most prokaryotes have a cell wall containing peptidoglycan, which is a special component made of peptide and type of sugar. Prokaryotes are believed to be much like the first cells that existed on Earth. So the general life structure of a prokaryotic cell, first of all, we see that the DNA is located in the nucleoid. As pre-mentioned before, prokaryotes do not have any individually wrapped components in it, which is why the DNA is kind of just jumbled around and just put in the cytoplasm over here. As you can see here, it just in the cell itself, and there's nothing else really there holding it together. So the ribosomes are also floating around the cytoplasm. The cell membrane is surrounded by a cell wall, which is why some bacteria are so hard to kill, because they have so much layers of protection. And the other structures shown might be present in some bacteria, but not all. So, for example, looking at the flagellum, which is kind of like a little tail that bacteria is used to move. Only some bacteria have it, and not every single bacteria have this feature, the flagellum. So prokaryotic cells are smaller than eukaryotic cells overall. The reason why they're smaller is because they're unicellular organisms, so they have to be able to survive on their own, which means the surface area to volume ratio is much more favorable for moving material in and out of the cell, because they have a relatively large surface area to volume ratio, which means for every bit of volume in this prokaryote, they have more surface area to deliver resources in and out of this cell for this bit of volume. However, the prokaryotes lack modifications found in eukaryotes that aid internal transport, because they are overall simpler organisms than eukaryotes. So this is just an elaboration on the surface area to volume ratio, that as cells get bigger, the volume increase faster than surface area. This is a simple example using cubes. I'm sure you guys have all played with this type of building cubes. So first of all, let's look at this single cube. The surface area is 1 because each side is 1, and 1 times 1 times 6 sides equals 6, and the volume is 1 cubed unit. However, if we increase each side by a factor of 2, which means now the side is 2 units long, and the surface area is actually 2 squared, which calculates the surface area of one of those little like two-dimensional shape of square. We actually have 24 units for the square units for the surface area. However, for the volume, we actually have 8 units cubed. And if we move on to side length equals 3, the surface area goes to 54, and the volume goes to 27 units cubed. So as you can see, even though surface area every time we're multiplying by 4, and we're multiplying number here, and the side length is increasing only by 1 per time, the volume is increasing exponentially. So in this case, the volume went from 1 to 8, and then we want from 8 to 27. And if we have 4, we'll actually have, I believe, 60 something cube. But anyways, as you can see here, the surface to volume ratio becomes smaller and smaller as the organism gets bigger. So next up is eukaryotic cells. Eukaryotic cells, this is the overall structure of eukaryotic cells. I hope don't get overwhelmed by this because we'll be explaining each part in detail afterwards. But the biggest component of it is there is a nucleus in the center. There are some endoplasmic reticulum around the nucleus. There are some other components fully around the nucleus that help the cell function, and last but not least, this entire thing is wrapped in a plasma membrane. So speaking of the plasma membrane, it's made of a phospholipid bilayer with embedded proteins. What I mean by the bilayer is that, as you can see here in the diagram, there's one layer on the top, and then there's one layer on the bottom. It's kind of like a piece of cake, I guess. The top layer and then the bottom layer. And each one of those little, I'll say they're like person-like shape. It consists of a phosphate head with fatty acid as the two tails, as you can see here. And what it does is that the phosphate heads are hydrophilic, so they suck the water onto them, but the fatty acid tails are hydrophobic, which means they hate water, and this is how the cells keep the aqueous environment from inside the cell separates from the aqueous environment outside of the cell. And in between that, there are some embedded proteins as mentioned here. And what the proteins do is that they kind of pump resources from inside to outside or outside to inside so the cell can survive. So for example, they might take in some vitamin, they might take in some food molecules that you ate, so the cell can use these molecules and build itself. The cytoplasm is the region between the plasma membrane and the nuclear envelope. So the cytoplasm consists of organelles suspended in gel-like cytosol plus cytoskeleton. We've mentioned about the organelles, which are the little compartments. And the cytoskeleton is basically the soft skeleton of a cell, and what it does is that it helps uphold the cell's shape so it doesn't all jumble up and it doesn't like fall apart or anything. So the 70 to 80% of the cytoplasm is water, but it's semi-solid because it has some other components in it, such as some proteins. So this is perhaps one of the most important part of a cell is the nucleus. There's usually only one nucleus per cell. It's usually the largest organelle in this entire cell. It's basically like, I guess, the headquarters of the cell, I guess, and it's bigger itself than most prokaryotic cells. While in other cases, the prokaryotes actually don't even have a nucleus. They have a nucleoid instead, which is the region where the DNA is held. However, in eukaryotes, which have nuclei, the DNA is actually being held in the nucleus itself. As you can see here, this big light purple region is the nucleus, and within itself, we have some tangles. They look like tangles of yarn, but that is your DNA in this nucleus. So nuclear envelope is the wrap that is wrapping around the nucleus. It's a double membrane envelope to ensure that there's no DNA getting out of the nucleus. It separates DNA from cytoplasm, and it helps separate some very important cell processes that we'll be talking about later on. There's some nuclear pores, which are like little holes that perforate this membrane. What they do is that they regulate the flow of molecules back and forth between the nucleus and the rest of the cell. So the nucleus contains an even smaller nexus called the nucleolus. The nucleolus is a region inside the nucleus where ribosomes are assembled from RNA and proteins, which we will mention in the rest of the slideshow. So keep an eye out for ribosomes. Oh, here. Ribosomes are made of two different size subunits. So they're actually slightly larger in eukaryotes than in prokaryotes. Noted that prokaryotes do have ribosomes as part of their cells. The ribosomes are made of special RNA, which are called ribosomal RNA, or for short, rRNA, and some proteins. They're all mixed together. So during protein synthesis, which is something that every single cell carries out, it basically makes protein that your body needs to function on. Ribosomes assemble amino acids into proteins. As mentioned in our previous lesson about biological macromolecules, proteins are made of individual amino acids. There are 20 types of amino acids, and they're like necklaces. When you link the amino acids, which are the little blue dots here, together, they kind of form a very long chain of protein that have a specific function. So in this case, as you can see, the ribosome is made of two parts. The biggest part is on the top, and then the other subunit is on the bottom. What happens is that this ribosome is going to read a strand of instructions sent from the DNA, and it's gonna be assembling the amino acid based on the purple instruction here. We'll be mentioning more of that later. Mitochondria, also known as the powerhouse of the cell. I think a lot of people refer to the mitochondria as that name. So it's a site for the conversion of stored energy to more useful form ATP. So what it does is that it converts glucose to something that your body can exploit and actively use in its cell. So the inner membrane of the mitochondria are folded. These, as you can see here, this is the mitochondria, and there are some I'll say folded walls in the center. The folds are called cristae. As you can see here, this is a cristae. And the area enclosed in the mitochondria is called the mitochondrial matrix. Basically, it's the rest of the space that is wrapped in this mitochondria. Peroxisomes are another relatively important component in the cytoplasm. However, I wouldn't say it's as important as mitochondria or the nucleus. But peroxisomes are small, rounded organelles enclosed by a single membrane. Note that it's not double membrane like the nucleus. What they do is that they digest and break down fatty acids and amino acids, and sometimes they even detoxify poisons, such as alcohol, which is quite poisonous to the body, which is why alcohol is technically not very good for the body. And they break down fatty acids that you have consumed, basically. So, contrasting animal and plant cells. So both animal and plant cells have microtubule organizing centers. Microtubules are basically what make up the cytoskeleton. However, animal cells also have something called centrioles, which are associated with cell division, and we'll be mentioning on the next slide. So animal cells each have a centrosome and lysosomes, but plant cells do not. Plant cells have a cell wall, chloroplast, and other specialized plastids, and a large central vacuole that animal cells do not. So chloroplasts are where photosynthesis happens. The cell wall is basically a very tough material that keeps the plant cells rigid, which is why when you look at a plant, it just doesn't like flop around. It's actually standing technically. And then we have a vacuole, which is where the plant stores its water and nutrients and salts, etc. So let's take a look at animal cells. This is an animal cell, obviously, as you can see here. The plasma membrane is not a cell wall. The plasma membrane is actually really soft, it's flexible. And it's important to see that here we have the secretory vesicle. So what happens is that the cell is going to intake some sort of, say, food from the outside environment. And what happens is that the food enters this little space over here, and the cell kind of moves around and encloses this food here. This vesicle gets sent up into the cell and into your system so your body can process it. And you can see here the nucleus here. mitochondria is the orange ones, and microtubule, like those little tubes over here, as well as these green strands and stuff, they all count as cytoskeleton, which is what is holding your cell up. And this is a plant cell. Perhaps two of the most notable things about plant cell is that they have a very rigid cell wall which maintains the cell shape. Usually it's squarish or rectangular-ish, and they have a huge, large central vacuole. In fact, when you look at it under a microscope, it looks like that the plant cell is being upheld by the vacuole almost. So it's filled with cell sap that maintains pressure against the cell wall. It also holds a lot of water. So, which is why when a plant doesn't have water, which means the vacuoles of the plant cells are now more empty, the plant wilts because there's nothing else to uphold the cell anymore besides the cell wall. So let's talk about centrosomes. Centrosomes are present in animal cells. They consist of two centrioles, which are shown below. These two little twister-looking, like the candy twister-looking segments. They lie at right angles to each other, and each centriole is a cylinder made of 9 triplets of microtubules, which microtubules are again cytoskeleton. So centrioles are especially important when an animal cell is dividing because the centrioles actually assemble. They send out some microtubules, and the microtubules help drag the cell into two. We'll be talking more about centrosomes and cell division in the upcoming lessons. So plant cell walls. The cell wall is a rigid protective structure external to plasma membrane. As mentioned before, it's very rigid. It's because it's made of cellulose, which is a type of carbohydrates, rather than peptidoglycan, which is what we see makes up like bacteria cell wall. So as you can see here, the blue lines over here, like they look like individual cells, like little blocks. And what is the outline of these blocks are the cell walls. So chloroplasts occur in plants. So chloroplasts are double membrane organelles. They have their own ribosomes and DNA like mitochondria. So both chloroplasts and mitochondria actually have a very unique origin that we'll be talking about soon. The inner membrane of the chloroplast encloses an aqueous fluid called the stroma. As you can see here, this is a diagram of a chloroplast. The yellow part is the stroma, which is the fluid. And it contains a set of interconnecting stacked fluid-filled membrane sacs called thylakoids. So as you can see here, I always see those thylakoids as like Mentos, the candy Mentos, and what happens is that each individual piece of candy is a thylakoid, and when you stack them together, they form something called a granum. So this is a thylakoid, this small circle, and this entire stack is called a granum. The plural for granum is grana, so there are multiple grana in this chloroplast. Talking more about the central vacuole in the plants. So it helps regulate water concentration under changing environments, and it contributes to cell expansion, which is why if you have hypertonic condition or if the plant is experiencing some dry conditions, we lose water, which is why it shrinks. As you can see here, this entire cell is kind of shrinked within its cell wall. Endosymbiosis. This is a note. This is very important, and it's how mitochondria and chloroplast were able to become part of plant cells and human cells, animal cells. So it is hypothesized that these two organelles actually originated as independent prokaryotic organisms. So basically, they were once upon a time bacteria. So they actually became endosymbiotes of the prokaryotic ancestors of the eukaryotes. So what happened? The scientists saying it's because, say, we have an ancient cell. This ancient cell was like some primitive form of eukaryotes. So what happened was that this eukaryote ancient cell consumed a mitochondria, prokaryotes. And what happened was that somehow this mitochondria did not get digested in the cell. It didn't get eaten, and in fact, it actually stayed inside the cell. And it was like, we could help each other. So the mitochondria ended up making energy for the cell, while the cell provided a friendly environment like a home for this mitochondria. In addition to having their own DNA and ribosomes, the cell of these organelles is actually similar to that of the independent prokaryotes. So this is very interesting, is because mitochondria and chloroplasts are basically like, I guess you could kind of say invaders from the outside, but since we have been living for them for so many millions of years, they kind of have adapted to our bodies, and they actually peacefully divide with our cells now. So a lot of research was undertaken by regarding the endosymbiosis relationship, which is when one cell consumes another and it ended up working together. And next, we're going to talk about the endomembrane system, which consists of internal membranes and organelles in eukaryotic cells that work together to modify, package, and transport lipids and proteins within the cell. So first up are the lysosomes. The lysosome in animal cells contain digestive enzymes. Digestive enzymes, when we mention this, you kind of think about your stomach. And yes, it is kind of similar. The digestive enzymes are able to break down the molecules that your cell intakes, and even we're not organelles. Sometimes they recycle old organelles, and what happens is that break them into smaller molecules that can be reused. It's kind of like the recycling bin of your cell. We have the endoplasmic reticulum, the ER. These are interconnected membranous sacs and tubules, as you can see here. Of course, this diagram is a cross-section of the cell. The endoplasmic reticulum, which I'll be referring to as the ER from now on, is this blue part over here. They're actually supposed to 3D. If you can imagine it 3D, it actually completes and it's like individual sacs, but in this case, they're linked together. So you can see it's kind of like a maze, and there's space inside this the ER. So what the ER does is that it modifies proteins and synthesizes lipids. The ER has two parts, the rough ER and the smooth ER. The rough ER is called the rough ER because it has those real red dots, which are the ribosomes on them, which are helping them to modify protein, and the smooth ER synthesizes lipids. So they kind of have different rules, but they are, after all, linked together, and what passes between them is kind of like this mutual process. So the hollow portion of the ER is called the lumen with cisternal space, and the membrane of the ER is continuous with the nuclear envelope, which means, as you can see here, this is a nuclear envelope, surrounds the nucleus, and it comes off and it's linked to this entire maze of ER. So as we have mentioned before, the rough endoplasmic reticulum is where a lot of ribosomes attach it to it, which is why it looks kind of rough on the outside. The ribosomes help manufacture proteins, and then this ribosomes spit the proteins into this ER, and the ER modify these proteins in its lumen, which is like the internal space. So then the modified proteins are either incorporated into cellular membranes or they're secreted from the cell, such as hormones. From the cell, they come out of the cell and they go into your body system to help signal a lot of stuff. So endoplasmic reticulum also makes phospholipids for cellular membranes. Phospholipids or modified proteins are not destined to stay in the RER, so they actually reach their destinations via transportation vehicles in your cells called vesicles. So basically the vesicles, as you see here, they kind of bud, which means they come out of the RER, and they kind of just deliver whatever got produced in the RER to the rest of the cell, which in this case, it looks like it's heading toward the Golgi apparatus. The smooth ER is continuous with rough ER, but it has few or no ribosomes on the cytoplasmic surface. As you can see here, this is a smooth ER. I would say it's a little bit slimmer than the rough ER, and it has no ribosomes like the red dots on it, which is why it has a smooth surface. So the function of the smooth ER is synthesizes carbohydrates, lipids, and steroid hormones. It sounds kind of like the functions for the rough ER. However, the rough ER doesn't synthesize carbohydrates or steroid hormones, but they both synthesize lipids. It also detoxifies medications and poisons, and it stores calcium ions. So I would say the first two bullet points are the most important things to know. The third one is really specific. If you can remember, do remember it, but I don't think it's that important as of now. Next, let's talk about the Golgi apparatus. The Golgi apparatus are lipids or protein within the transfer vesicles that are still needed to be sorted, packaged, and tagged so they get to the right place. So basically, the Golgi apparatus is where this entire process happens. It's kind of like the post office. So I have a package that I want to be sent to, say, my distant relative. So I bring this package to the post office, which in this case is the Golgi apparatus, and then I will ask the officers at the post office to help me wrap it around, to help me put a QR code on it and the address and all the information for my aunt on it. So this is what the Golgi apparatus basically is. They tag this package and then they send it out to the other phase. So as you can see here, this is my package. It enters the Golgi body from here, and then as we go through all the sacs in the Golgi body, it comes out from here, all packaged, and it's all labeled. And then because of the label, your cell is able to know where to deliver this, whether we should throw this out of the cell and into the body system, or if we should keep it or like send it somewhere else. So to go into more details, so I don't think you necessarily need to know this, but just extra information. The receiving side of the Golgi apparatus, which is where I put the package in, is called the sys phase, and where the package comes out, like the finished package, is called the trans phase. So there's transfer vesicles from the ER fuse with the sys phase, empty their contents into the Golgi apparatus, and then come out of the trans phase, where they are once again wrapped in membrane. And this often involves adding short chains of sugar molecules, which are kind of like ID cards, or in this case, like QR codes that we add onto the package. So the cell will be able to identify, oh, I need to send this package to the other end of the cell, or I need to send this package outside of the cell. The cytoskeleton is basically like the skeleton of your body, except it's much softer. It's a network of protein fibers with several functions. There are many types of cytoskeleton. Their main role is to maintain the shape of the cell. However, they sometimes also link organelles together and hold them in place so they don't float around randomly inside the cell. However, they still allow movement of cytoplasm vesicles within the cell. They don't really restrain them. They just enable cells within multicellular organisms to move in some case, such as white blood cells. So there are three main components, say types of cytoskeleton. First one are microfilaments, then we have intermediate filaments, and last but not least, we have microtubules. They all have different sizes and different functions. For example, microtubules, as depicted here, are used in cell division. Microfilaments are used to kind of pump the cell up, like to strain it and really divide it and like exert force on it when the cell is dividing. And intermediate filaments are used all around the cell to hold the organelles in place. So microfilaments are involved in movement of the whole cell or internal parts, and they determine the stabilized shape of the cell, and they're made from active monomer. Microtubules form rigid internal skeleton from some cells. They provide framework for motor proteins to move structures within the cell, and they're made of tubulin dimers. I don't think you have to know the third bullet point. It's a lot of memorization, but just know that microtubules is relatively rigid, and it's like the framework. And we also have cilia and flagella, which are similar to cytoskeleton, but they're not really cytoskeleton. Not every cell has cilia or flagella. So basically, they're also made of microtubules, but cilia are like shorter and more numerous. And flagella is usually just one single long strand. It's kind of like a whip. They're actually a flagella on sperm cells, but they also beat at a different rates and patterns. But both of their function is to either move stuff along the surface of the cell or to move the cell in the body, and they're outside of the cell's membrane. So extracellular structures, just more on those. As mentioned before, there's the plant cell wall, but there's also some extracellular structures in animal cells as well. I actually did not know about this, so this is very interesting. There are three main components: collagens and other fibrous proteins. If you look at a cell diagram, you can see some diagrams have labeled those kind of elements floating around the cell to keep the cells from mixing, I'll say run into each other. There's also glycoproteins, and there's some linking proteins. So here's an example of what exactly is outside of a cell's membrane. Intercellular junctions provide direct channels of communication between cells, and plants and animals do this differently, as we can see later. So the plant cells communicate with each other through something called plasmodesmata. There are channels that pass between cell walls in plants to connect cytoplasm and allow materials to move from cell to cell. So in this case, as you can see here, this little channel here is called a plasmodesma, or plasmodesmata. And you can see the cell wall here, cytoplasm here, and what it does is that it connects these two cells together and forms a pathway between them. Because cells need to communicate with each other, because cells release chemicals such as hormones into the environment that they will like the cell, their neighbors to know about. And there are also tight junctions. Tight junctions are watertight cells between animal cells that prevent materials from leaking between cells. So these are found in epithelial cells, which are skin cells, that internal organs and cavities, as well as internal organs and cavities. As you can see here, these are a tight junction, and what happens is that they're in between the cells, and they basically prevent the cell from leaking into each other, because cells are after all aqueous environment, and it could be relatively risky if one cell does rupture and the content in the cell comes out. There's also desmosomes, which are short proteins in the plasma membrane that act as spot welds between the cells. So it's kind of like jigsaw puzzle, I guess, where you can kind of click the two pieces together. So these actually desmosomes join the cells together in tissues that stretch. This is very important in organs such as heart, lungs, muscles, etc. They're constantly moving, they stretch a lot, and we don't want the cells to simply disconnect from each other. Imagine you're stretching and then one of your tissue just falls apart. We don't want that to happen, and that doesn't happen thanks to desmosomes. They're only present in animals. As you can see here, this is the desmosome, and what happens is that they kind of interlock with each other, and these two are, I believe, the cell membranes of two different cells. So last but not least, we're going to talk about gap junctions. They resemble the plasmodesmata in plants because they also form channels, and they allow signals and atoms and ions to be moved between cells. They develop when 6 proteins form like a donut or flower-shaped structure in the plasma membrane, as seen here. And when these little flowers from 2 different cells line up, it completes the channel, which means, as you can see here, this is the cell membrane of one of the cell, this is the membrane of the other cell, and when the flowers line up, the channels, you can now communicate between these 2 cells. So the gap junction is a protein line pore that allows water and small molecules to pass between adjacent animal cells, and that is basically the main idea of how cells communicate in between each other. And this is very important because there are certain body conditions. In fact, there's a lot of signal traveling through your bodies right now. And if cells cannot talk to each other, they wouldn't be able to perform functions together effectively. For example, cells in the same tissue or the same organ have to be on the same page when they're performing a reaction or, like, say, digesting food. And we have to have them work together instead of working on their own. So basically, I hope this slideshow has been informative, and I hope you have learned more about how the cells function. And it's important to keep in mind that you are made of cells, and it's so intriguing to think about how there are so many cells in you, and each of them is so complicated in their own unique ways, and just how much work it requires. And anyways, this lesson just makes me really grateful for myself and my biological functions, because it makes me feel like I am such a multicellular organism that has so many functions within me. Anyways, thank you for listening. And if you have any questions, please feel free to ask me at this email. Type in the speaker's note, quilling.wu@outlook.com. Please do not hesitate to reach out. I would love to help you. I have taken biology, and please, yes, let me help you. Thank you very much, and I hope to see you guys next Saturday.